So we already discussed teeth and gums, how they are important for the beginning of the digestive process. What is it called when we chew? Mastication. And what do we masticate? Food into a bolus. What's a bolus?
Spit and food.
Exactly. Saliva squirt from salivary glands throughout the or surrounding the oral or buccal cavity, parotid, sublingual, submandibular, salivary glands, all shooting, because what kind of glands are they? Yes, they are, but what kind of gland am I? An exocrine gland, so I shoot through little tube-like ducts into the oral buccal cavity. So, mastication, chewing into a bolus, ball of food. Important. Why is chewing important? Yeah, you remember, the whole digestive process is taking big things and making them good. So we have to chew to start the process. The saliva is going to add fluid. That's helpful because everything, chemistry, needs some sort of liquid medium to happen at an efficient rate. Also, what's in saliva that's going to help with the digestive process? Enzymes. Enzymes are going to play a big role in the digestive process, as we see today in lecture. And then what do I have to do with my bolus of food after I've masticated it? No, I don't swallow it. I deglutition. Of course, which is swallowing. Now, when I swallow it, something happens that's going to help me out respiratory system-wise. Yeah, when I swallow, those tongue muscles and those posterior muscles in the mouth are going to pull down and cause the what to close the trachea shut, that epiglottis we met in the respiratory system. And then food's just going to fall from the mouth, boom, into the stomach. No, what has to happen? Peristalsis, and that is rhythmic contractions of this. What is this? The esophagus, to then take that bolus of food, throw it into the stomach, where it now will become something new. We're not there yet, but of course you read because you were excited. It becomes something called chyme when it mixes with the gastric juices of the stomach. So, know those fancy schmancy terms for all of this stuff. And let's pick up on this tube. Well, the pharynx. We got to go by the pharynx. We shared the pharynx with somebody else. Respiratory system. What part of the pharynx do you think is behind the mouth? The oropharynx. And then the laryngopharynx will feed into the respiratory trachea. And then we're going to take a bypass after we've gone past the oropharynx into the esophagus. So, the esophagus has all the same layers that we saw before with the tube. Remember, the tube is the same all the way through. We're going to have different types of cells and different thicknesses of layers, but we're going to find all the same layers throughout the tube. So we're starting with the mucosa, the submucosa, the muscularis. And then the adventitia is what? Kind of wrapping everybody up, holding everything together. And we're going to find it here in the esophagus as well. So what kind of cells am I going to find lining the lumen of the beginning of the digestive system tubes, the esophagus? Lining the lumen. There we go! Stratified squamous epithelial cells. And this is where we're going to see one of the biggest differences in the lining of the lumen. Okay, so why do you think I have stratified squamous epithelial cells in the esophagus? But rest. To prevent damage. It's protection. It's like your skin. If I didn't have a stratified layer out here, I went like that, and then I'd be into my body, right? When you swallow, You stretch that out. That's not stretched out. That's kind of all clumped up together. What's going to happen when you swallow a Twinkie? It's going to expand, and that's going to rub down the sides. Or when you swallow, what are you eating? Donuts? Yeah? Little powdered ones? I noticed, sorry. So when you eat powdered donuts, what's going to happen? It's going to stretch out, and they're going to rub down the sides. So that's a protection mechanism. If we didn't have that, we wouldn't be able to swallow very much. So that's going to stretch way out, and those are the layers of the esophagus. There's also glands in there that are going to help things move along in the digestive system. And what kind of glands do you think we have? Anybody? Yeah, and mucus-like glands. We're going to find a few of those in the esophagus too. We're going to find a lot more of them down later when we hit the parts of the tube like the stomach and the small intestine, large intestine. We'll find a few of them in the esophagus as well. So what is this? What is this diagram showing us? What the heck is it, by the way? Oh, there it is. Page 867. This is the process of swallowing. So, the tongue, the mouth, the saliva, all that good stuff we're talking about in the upper esophagus, the sphincter in the upper esophagus, which is what they talk about in the textbook, but it's nicely outlined here in this diagram, and you know me, I like my diagrams. So, upper esophageal sphincter is contracted, and that's what's going to happen, or that's what's going to help happen to do what? Swallow, push stuff down. So, this is the first part. Tongue presses against the hard palate, forces the bolus of food into the oropharynx, behind the mouth, and then the involuntary phase begins. So, everything else before that is voluntary. I know I'm doing it, I'm making myself do it, but once I get past that point, then everything else is involuntary. That's why I have to shove the pills down my dog's throat, because the dog's not going to do the whole voluntary part. No way, no how. But once I get it past that point, then involuntary reflexes take over. I know it sounds disgusting, but I did have to give her a couple of Peptos this morning. So, food bolus is sent down. This damn thing. All right. So, uvula, and that's that little hanging thing on the back of your throat. Larynx rise to prevent food from entering respiratory passages. This is when my nice epiglottis does its trick. And the esophageal sphincter relaxes now and allows food to do what? Now it can go down into the esophagus. And then we have peristalsis taking place with the help of constrictor muscles in the esophagus. They're going to help push that bolus of food down into the stomach. So, what's a sphincter muscle? We met some in the circulatory system. We had precapillary and postcapillary sphincter muscles. Yeah, they're little muscles that surround tubes, and when they're constricted, they do what to the tubes? They close them off. We're going to meet a whole bunch of them in the digestive system because we want food to be in certain portions of the tube for a certain duration of time in order for the digestive process to take place at an efficient rate. And those sphincter muscles are going to help close off or section off parts of the tube as we go through the digestive process. So food is moved through the esophagus to the stomach by peristalsis. Has to get by what in order to get into the stomach? It's got to get by a sphincter muscle. So pressure of the contraction of peristalsis is also going to play a role in opening the gastroesophageal sphincter, which is the connection, that sphincter between the esophagus and where all the gastric juices are, the stomach. So see where the name comes from? Gastroesophageal, and it's a sphincter, so sphincter is added to the term. So the gastroesophageal sphincter opens and food enters into the stomach. What do you think is going to happen after the food goes into the stomach? The sphincter muscle is just going to stay open. No, it's going to close shut. Now sometimes people have problems with hernias in those areas. Maybe their gastroesophageal sphincter doesn't snap shut the way it should. And what might happen? Things that are down in the stomach region, which are a hydrochloric acid bath of juices, can wash up past the sphincter muscle and cause problems with esophageal erosion. What's that called? GERD, gastroesophageal reflux disease, or reflux, acid reflux. Anybody ever hear of that? Okay, so it's a problem sometimes with that. If I have something called a hiatal hernia, anybody ever hear of that? Okay, sometimes that can cause problems with that sphincter muscle closing properly, and you might run into problems with GERD. So they describe it in the esophagus as heartburn. Why do we call it heartburn? It's just right around here. Where does the esophagus go? Physically. Runs down behind what? Runs down behind the trachea. It hugs it tight, which runs behind the heart. So until we get into the bronchioles with respect to the trachea, then the esophagus is on its own to go down and hook up into the stomach. That is going to happen right. We have to have a little pass through the diaphragm before we get into the stomach. So that's right in here, and that's why we call it heartburn. Okay? So they talk about a hiatal hernia and heartburn under homeostatic imbalances under the esophagus. And then, of course, me and my steps, I like the whole learn to swallow thing. That has 5 nice steps. Who knows? Nice essay question. And then the digestive process, which is kind of already started in the mouth, begins. So the digestive process, mouth to esophagus. Here's the terms we need to know. Mastication, chew it up. What else am I going to do? Spit on it to create a bolus. Then I'm going to swallow it, deglutition, and that is then going to go into the stomach. So now we're into the stomach. The gross anatomy of the stomach is a little bit different than the gross anatomy of the esophagus. We still have all the layers, don't we? We have a muscularis externa, but this time we have a whole bunch of different muscles going in a whole bunch of different directions. We had a few going in different directions in the esophagus, but not as crazy as it is in the stomach. Why do you think? What's going to happen in the stomach? I'm going to do 2 major breakdowns in the stomach. 1, chemical. 2, mechanical. By doing what? Yeah, exactly. So contractions of all those muscles going in many different directions are going to continue that mechanical process by churning the food in the stomach so that it mixes really well with those gastric juices that we're going to produce there. So we see them outlined as the longitudinal, the circular, and oblique layer. And the beginning portion where the esophagus kind of hits the stomach, that's called the cardia. Did you ask yourself that question when we made you look at slides? It said cardiac stomach. What the heck is that? It should really say cardia stomach. And that's just where that particular slice of tissue came from, that region in the stomach. And then we kind of bump up and create something called the fundus. Then the longer portion of the tube, and now this is still the tube, it's just fatter. Yes? we have the body, which is the major portion of the stomach. Then down, as we come lower on the stomach region, we have a greater curvature. And then before we enter the next portion of the tube, which is what? Small intestine, we have a region called the pyloric region. So the pyloric antrum and pyloric canal. And of course, we have another trap door there, another what? Sphincter muscle. And what do we call that sphincter muscle? The pyloric sphincter muscle, because it's in the pyloric region. Now that stays shut for a period of time so that what can happen to your food after you deglutitioned your bolus after mastication? It's going to stay in there and get churned and turn into chyme by mixing with gastric juices. And that door is going to stay shut, and it's based on chemical messages that the body's going to get sent, and that's going to be a little bit of a confusing portion of this chapter, one of those psychotic diagrams we're going to have to look at again. It's over on page 872, figure 23.17. But these are all the chemical messages that are going to help with the digestive process. And there's some of these chemical messages that are also going to help control the trap doors. The pyloric sphincter being one of them. Okay, so we're going to get into that in a minute. But, so here we have the stomach. Now, beautiful, isn't it? That's a real one. You have to kind of pull out the spleen with it as well. Who's it sitting underneath? And you're going to see all this when you start your pig dissections. Because guess what? You kind of set up the same way as a pig. And it's cheaper to dissect a pig than it is to dissect you. We have more space to store them. But you set up the same way. So what you're going to see when you do your dissections is you're going to see the stomach hangs out right underneath the liver. And again, we can see the portions of it. The fundus, the body, the greater curvature, the lesser curvature on the inside, and then way down at the bottom there, we see where the small intestine begins with the pyloric region. All right, let's have a peek at the inside, because the inside is different. The inside is different from the inside of the esophagus. So now the lining of the lumen of the stomach, we're going to have a different description. Does everybody understand what I mean? Still the same tubes, still the same parts. Submucosa, muscularis, serosa. What's wrapped around the outside? Adventitia, yes? But they look different now. So when we look at the the muscle layer of the stomach, we still see muscles going in different directions. Again, we're kind of on steroids when we're in the stomach with muscles going in different directions. Because remember, what do I have to do in the stomach? A lot of squeezing and churning. So we have the submucosa, and take a close look at the inner layer called the mucosa. Different than what we saw in the esophagus. What kind of epithelial cells line the mucosal lumen of the stomach? And are they stratified or are they simple? They're simple. Because now we have to pay more attention to chemistry. We have to pay more attention to secretions, because this is what I have to do in this portion of the tube. So, if I have a stratified layer, things can't typically pass across a stratified layer, can they? Because that's kind of like a protective barrier. Think if I had a stratified layer of cells in the alveoli. Would that work very well? No. So, what we see here is a simple layer of columnar cells. And dotted throughout that simple layer of columnar cells, we're going to find them arranged a little bit differently. So they're not flat like the stratified squamous cells. They kind of invert themselves a little bit. to create something called a gastric pit. And inside the pit, guess what I'm doing? I'm making all sorts of good stuff, creating those gastric juices. And that's going to happen down in the gastric gland. In the gastric gland, we're going to be introduced to several different types of cells that make several different things. But right above that gastric gland, we are going to find something called a mucous neck cell. So there's a bunch of mucus being produced right above that gastric gland. Why do you think that's so? Yeah, it's for protection. It's for protection of the lining in the rest of the stomach. Because that stuff that we're going to talk about that we're making down in the gastric gland is made to break down substances that you put into your mouth. It doesn't know the difference between substances you put in your mouth and you. Because a lot of the substances you put in your mouth, believe it or not, are very similar to you. Right? So it doesn't know the difference. It's just a chemical. It doesn't have a brain or a nervous system or anything like that. So we have to protect the cells that line the lumen of the stomach by making lots of mucus for protection. So down in, and we start talking about these guys on page 870, down in the gastric gland, excuse me, we're going to see several different types of cells. So again, the mucous neck cells there for protection. The parietal cells, what are they doing for me? And the parietal cells are those nice looking, I don't know, they look like little Pac-Man cells. There's a bunch of mitochondria in there. And what do they, bless you, what do they do for a living? They secrete hydrochloric acid. What's that for? Yeah, chemical breakdown. If you take hydrochloric acid and throw anything in it, you're going to see stuff happen right away. So it's a very volatile chemical that'll help with the breakdown process. The other thing that hydrochloric acid is going to do for us is it's going to add a bunch of hydrogen ions to this mix that's going to help with breakdown of substances. The other group of cells we have are things called chief cells. And parietal cells and chief cells, they kind of work together because chief cells produce substances that are going to help do what? Start breaking down stuff. Particular what? In particular what? Proteins. And what's the substance that those chief cells are going to produce to help me break down proteins? Pepsinogen. Now, pepsinogen, in order to work well, needs some acid. Who's going to help with that? The parietal cells, to make something called pepsin. And that's going to help with protein digestion. So that's another thing that we're making inside the gastric gland. And then they talk about something called an enteroendocrine. Endocrine. Endocrine. What's that make you think of? Huh? It makes you think of it. What else does, what was that kind of thing that drove you crazy that you had to memorize all of that stuff in the endocrine system? Hormones! Yes? So the enteroendocrine cells, and what the heck do I need hormones for? This is digestion, for goodness sake. What are the hormones going to do for me? What do hormones do? And cause cells to do what? Something different. So I'm getting hormonal messages also being sent through the enteroendocrine secretions, and we can see them over on page 870. Some of the secretions we're going to see are histamines, serotonin, somatostatin, gastrin. All of these are going to play a role in signaling the next portion of the tube. Because everybody kind of needs a heads up as to what's coming. Your mouth gets a head up from your brain. You watch the Pizza Hut commercial. You watch the Burger King commercial, or somebody's in the kitchen cooking. Yes? That's the heads up for the next part of the tube. The next part would be your mouth. What do you start doing when you think good stuff's coming? You start to secrete that fluid. That's the heads up. That's the cephalic phase of digestion. That prepares the next part of the tube for what's to come. You chew, you swallow. That's the heads up for who? Something's coming down. The stomach. Okay? And the stomach has to give a heads up to the next part of the tube. And what's the next part of the tube? The small intestine. So part of that heads up, hey, something's coming, is the secretion of these hormones. That's going to prepare the small intestine for what's to come. Because soon that trap door is going to open, and the chyme is then going to flow from the stomach into the small intestines. Small intestine is going to be ready for that. So the enteroendocrine cells are going to help with some of the hormones that are being produced. Now, if I don't make enough mucus, I'm going to start eating my own stomach away. I'm going to start to digest my own stomach. What's that called? It's called an ulcer. Now, that can happen in different places of the stomach. In that pyloric region down the bottom, that's one of the more common areas we might develop an ulcer. And you can see under homeostatic imbalances on page 870 and 871, their discussion of ulcers. Peptic ulcers, gastric ulcers. There are ulcers in different areas. And an ulcer is basically a wearing away of that inner lining or lumen and exposing a whole bunch of nerves. And why else might that be dangerous? You might get things into the circulatory system that you don't want to get in there, and then you might even leak some of that stuff out into the outside cavities around the stomach, which would be very, very dangerous and could cause sepsis. What's sepsis? Yeah, that kind of spreads throughout. And remember, I'm below the diaphragm now, so I'm into what? I'm into the abdominopelvic cavity, which is a pretty darn big cavity with quite a few different organs in there. So there's a bunch of fluid in there floating around between all of these wrappings and organs. And if I dump stuff into that, it sort of spreads throughout and around all of those organs, and that can be very dangerous. They also talk about and have shown a nice picture of on page 871, a nasty little critter, which is kind of new on the scene. We thought, no way can bacteria live down there in that hydrochloric acid bath. Well, we were wrong. There's a nasty little bacteria called the Helicobacter pylori, and it has been associated with the formation of ulcers in patients. Hangs out in the mucosal layer of the stomach and can cause issues with that mucosal layer. Kind of slow down your production of mucus. And again, mucus is your protection layer. So we don't want to slow down the production of mucus by any means. Okay, so digestion takes place in the stomach. And here's that nasty diagram. This nasty diagram is going to tie everything together for you. So it's your visual for tying together all the different phases of the digestive process. So we've already started talking about the cephalic phase. Now we're into what phase? When we hit the stomach, we're into what phase? That's the gastric phase. In the gastric phase, we're going to continue with production of chemicals and hormones. And again, in this diagram, you can see where some of those hormones are going to play their role in getting the next phase of digestion, which we're going to hop into once we leave the stomach, which is the intestinal phase of digestion, ready to go. So we see on the left side of the diagram, a lot of the actions that we see described here are stimulatory actions. They're preparing the next parts of the tube for what's to come. And then we see that we also after things have passed that portion of the tube, we need to relax that portion of the tube. So we have inhibitory factors controlled by some of them by the central nervous system, but also some of them are controlled by the hormones that are produced by the enteroendocrine cells, slow down that portion of the tube after its job is done. So look this over. It's good. Ties everything together. So in your textbook, you'll see regulation of gastric secretion. You, again, will see description of the cephalic phase. Then we see a description of the gastric phase. And again, these chemicals are going to help to break down further large things into small with the help of hydrogen ions produced by the hydrochloric acid. And which cells produce those again? Parietal cells. Chief cells are producing what? Pepsinogen. Together they're going to be able to form pepsin, which is going to start protein digestion in the stomach. And then again, we're physically making things smaller with mechanical digestion in the gastric phase. We're going to get to a certain pH, a certain point, and that's going to be the trigger for, what was that sphincter muscle called again? The pyloric sphincter to open. And once it opens, we're going to then toss what we have, that chyme, the mix of food and gastric juices, into the third phase, the intestinal phase. So what kind of cell is this? That's a parietal cell. So on page 874, we see the mechanism of action that hydrochloric acid is going to set forth. And all of these ions are important for the breakdown of food. So this is the stomach lumen. This is the parietal cell. And some things are actually going to pass from the stomach into the circulatory system. One of the major things is a buffer called bicarbonate, or HCO3. And bicarbonate ions are a major buffer system in the body. What's a buffer system? Yeah, it's a pH regulator. Because remember, pH has to be very tight throughout the fluids in our body system. What's a normal blood pH? 7.35 to 7.45. So right in the beginning in this digestive process, we're going to contribute quite a few bicarbonate ions to help with buffer systems in the circulatory system. Another thing that we're going to help make is chloride ions, which are also a major portion of the buffer systems in our bodies as well. So we have a bunch of chemical reactions going on in the stomach to help regulate the digestive process. Again, the concentration of these ions is going to help regulate what goes on in the stomach. So on page 876, we see peristaltic waves in the stomach. Some of these chemicals are going to help with that as well. To help with propulsion, moving things from one place to another. The grinding process, which is vigorous peristalsis, and the mixing action that needs to take place while the pyloric sphincter muscle is still closed. And then once we get chemically to a certain point, when the pH is just right and we have enough ions and we have enough hormone from the, what were those cells that made the hormones in the gastric pits? The enteroendocrine cells. We'll get to this phase called the retropulsion phase. And that's going to do what for me? That's going to help open the trap door. Again, don't forget the chemicals that we just discussed are also going to set up the small intestine for what's to come. Now the pH of the stuff the chyme that goes into the small intestine is very what? It's very acidic. It's a very low pH. So we're going to talk in a moment about trying to kind of even that out, because the small intestine doesn't want the pH to be that low. So I'm going to have some help from other portions, not the tube itself, but other portions of the digestive process. And what am I talking about? It's not the tube, but there's other things that help with the digestive process. They're called accessory organs to the digestion process. Things like the liver and the pancreas. And they're going to help even out that pH. Now, typically, because of the process we just discussed, things move in one direction. But they describe something not moving in that direction under homeostatic imbalances. What's that? So when the muscles kind of back it up into reverse and things go in the wrong direction. What's that? What do you do when that happens? You vomit or emesis. An unpleasant experience, if you've ever experienced it, for sure, but chemical imbalances can cause this. Sometimes aversions can cause this, cause you to sort of back up those muscles and have them go in the opposite direction. So, what's this? On page 877, figure 23.2, describes neural and hormonal factors inhibiting gastric emptying. So remember, I want to hold on to this stuff in the stomach for a certain period of time so I know that mixing has taken place properly. So, presence of fatty, hypertonic, acidic chyme in the duodenum is going to cause what? Follow that dotted line down there. Yeah, it's going to slow the stomach down. So that's one of the signals for the next part of the tube. So, when I start to empty, when that pyloric sphincter starts to open slightly because of chemical changes and starts to dump things into the beginning of the small intestine, and what do you think the beginning of the small intestine is called? It's called the duodenum or duodenum. That's going to start slowing the stomach down a bit. So, all of the other controls in there cause the stomach to do its thing. Central nervous system, increased sympathetic, decreased parasympathetic activities will eventually slow the stomach down. Enteroendocrine or enteric neurons that pick up those hormones that we secreted are also going to help slow the stomach down. And then some of the hormones that were secreted by those enteroendocrine cells are by the stomach region of the process. Things like secretin, cholecystokinin, vasoactin, intestinal peptides. All of these are hormonal-like signals that will eventually slow the stomach down as well. So again, we have to build them up to a certain point before the trapdoor will open and we can start the next process. So once we get past this, we're going to empty into what? We're going to empty into the small intestine. So first I talk about gross anatomy of the small intestine, but since we have this diagram up here, we might as well talk about these guys first real quick. The small intestine starts here. So what's attached to this? Everybody's in a coma today. The stomach. What the heck? Oh, I can't do the pen because I don't have my PowerPoint. So the stomach's over here, yes? Remember, chemical signals, we're going to open that trap door to start things coming down here. Those chemical signals are also going to signal the accessory organs of the digestive system. And those guys are the pancreas here and the liver here. Now, the liver makes a very important substance to help with the digestive process. Anybody know what that's called? It's called bile. The liver makes it. But it's got a nice little storage tank down here because it makes it kind of slow and it's kind of watery. So it's got a storage tank called the what? The gallbladder. The gallbladder is going to store that bile as it's produced by the liver, and it's going to concentrate it, kind of suck the water out of it. Just like your concentrated laundry detergents. What is that? Yeah, it's a lot less water, just more soap. In the old days, you'd have the giant jug with the same amount of soap in it, but more water. So you'd have to add more of the substance to your laundry to get the laundry clean. Are you adding more soap to your laundry with concentrated laundry detergent? No, you're adding the same amount, except you only have to add a little bit because we did what? Sucked out all the water. And that is the function of the gallbladder. So that I can then, with that chemical signal we just discussed, when things start pouring into the small intestine, I can get my cue to do what? Throw in some bile. Along with, and when we talk about the pancreas, we'll talk about the secretions, along with secretions from the pancreas that are going to help the chemical process of digestion as well. They share a common tube. Anybody know what that's called? Yeah, it's called the common bile duct. And sometimes, because of the whole concentration process, what might form in the gallbladder? Yeah, crystals. And those crystals we know fondly as gallstones. Is that a problem? Yeah, it can be, because those little rocks or crystals or stones can get caught up in the what? Can get caught up in the tubes and cause problems with backing up. Anybody have their gallbladder removed? No? Everybody's got their gallbladder in here? Good. Anybody have gallstones? No? I do. They're good, though. We're good with the gallstones right now. We're living together in peace and harmony, so I have to be careful. But if people have a problem with the formation of gallstones that actually start blocking up their tubes, you can go in and remove the gallbladder, but what do we keep intact? Yeah, we leave the bile duct alone, that common bile duct, because liver is still going to make bile, and it's still going to shoot it down the chute. What is bile for? Doesn't break down anything. It emulsifies. What's the difference between breakdown and emulsify? Well, I'll give you a little visual. If I take and try to make salad dressing, oil and water, yes? What are the lipids going to do? They hold on tight to each other and they repel water. They float to the top, don't they? They are what? Phobic. So if I add an emulsifier to that, and you did it in lab, what was the emulsifier you used in lab? Bile salts. So what happened when you added the bile salts to the tube? In the tube that you didn't add bile salts to, you shook it up and whoosh, everybody separated, right? But when you added the bile salts, what happened? It caused those hydrophobic molecules to kind of lose their grip a little bit on each other so that they could go into that solution a little bit easier. So it breaks apart. Pretend my hands are lipid molecules. It breaks them apart. Now, who's going to break them down? not bile. Our friends, the what? You should know this if you have a lab exam tomorrow. Oh, today. You have it today. Who's going to break down lipids? Lipids are made of glycerol and fatty acids. Who's going to break them down? Thank you. Lipase, an enzyme. See the difference? So bile is going to break the hold, and lipase is going to break apart. So we scored some of that right at the beginning. Now who makes lipase? You should know that too because you have a lab exam today. Not the liver. That little other accessory organ. The pancreas! So right at the same time, I'm going to give a nice squirt of pancreatic juices, which contain several different enzymes, to break apart some of the different molecules that we're talking about. We're also going to get a bunch of enzymes secreted by who? Besides the liver and the pancreas, who else makes enzymes to help the digestive process? The small intestine. What are those enzymes? There's a whole bunch of them, and they group them into a group of enzymes called the brush border enzymes. And this is where we start getting down to the nitty-gritty of breaking things small enough so that I can do what with them? Absorb them into cells. So now we're talking about molecules that are small enough to use at a cellular level when we enter the next portion of the digestive system. What time is it? 9:30. Oh, I got plenty of time. I feel like I've been talking a long time. All right. So this is the lining or the lumen of the small intestine. Now, it looks different than the lining or the lumen of the stomach. Still part of the tube, so we have all the different layers. The lumen, the mucosa, the submucosa, the muscularis, the adventitia, they're all there. But now things look a little bit different. One of the things we're going to find is that the submucosa, the adventitia, are going to be much more highly vascular in the small intestine than they were before in the tubes. Why do you think? Yeah. Now we're going to get down to business as far as food being, or molecules being absorbed. So we're going to find that the adventitia in the small intestine region, and what do we call that? That had its own special name. Have we learned that yet? A term called the mesentery. I think we learned that. I think we did. Remember that term? Mesentery? It's kind of when we were talking about all the layers and way back in the beginning. Okay, the mesentery that holds all the small intestine. Remember I said you had to hold them together because there's 21 feet. You got to kind of hold them all in there. There's membranes that hold them all in there. That's the mesentery. And the mesentery is going to be very highly vascular, and we'll see that when we dissect our pigs. One of the fun things, if you're in my lab, that you get to do is take all the parts out at the end. And one thing I want you to pay, even if you don't get to do that in your lab, I want you to pay close attention to the small intestine and the membranes that hold all the small intestines together. You can actually pull them apart just a little bit, and it looks like a fan of blood vessels. So you'll see a whole bunch, because we get you pigs that are injected, you'll see blood vessels, a whole bunch of little blue and red tubes inside that mesentery. So that's what they're depicting up here in the diagram. So when you absorb things from the small intestine, it has to make its way through where before it goes to the rest of the body? Anybody know? I'll just give you a hint. Where do you think the hepatic portal system goes? The liver. So we have to pass through the liver first. Liver, among its many, many jobs, one of the things the liver is going to do for me is help me process lipids, and it's also going to help get rid of toxins for me as well. So I have to pass through there first. Those macrophages in the liver, and I think we've mentioned them before, but they're called Kupffer cells. They're going to help filter out any nonsense that I'm absorbing from the small intestine, and again help with the beginnings of processing of lipids. Now, lipids aren't going to be in the circulatory system first. They're going to be where first? We talked a little bit about this when we talked about that other set of tubes we have, the lymphatic system. Okay? So, the inside lumen, no more gastric pits. I don't invert my lining. I open it up. And I form these finger-like projections with cells that line them. Now, why do you think I do that? More surface area. So if I were able to, and I can't physically do it, but if I were able to now take this and stretch it out flat, it would be about that big. But it's in this much space. So see what I just did to my surface area? I gave me a lot more room in a much smaller space to do what I need to do in the small intestine, which is continue to break down substances and to, most importantly, absorb those substances into the circulatory system. So these things are called the villi. Let's take a peek at them up close and personal. So these villi, these little finger-like projections, running up through the center of them are going to be what? What's those blue and red tubes running through the center of my villi? Circulatory system. They're capillary beds. But what are those green tubes? Lymphatic system. They're called lacteals. And those guys are going to help me do what for the digestive process? Absorb what kind of nutrients? Most of the other stuff goes into the circulatory, so it's small enough, we're good. But some molecules are too big. Know anybody that's too big? Lipids. So lipids are going to be able to go into the circulatory system. Remember when we talked those little flap-like valves that the lacteals had? Lipids that are larger can fit into those. So how do lipids get into the circulatory system then? I know they're there. How do they get there? If the lacteals are going to absorb them first, how do they get to the circulatory system? Do the who? Where? Point. Yes? Remember the right and left thoracic duct dumped into the circulatory system? So all of that lymph, which is going to contain from the small intestine some fat, will dump back into the circulatory system that way. So that's how the lipids get into the circulatory system. You kind of have to take a detour through the lymphatic system as well. So if I look at the lymph fluid, it's not going to be as clear as blood plasma because it also has a bunch of lipids that it's carrying along with it as well. So it's more milky looking than plasma. So the lining or the lumen of the small intestine, different. We have the villi. What kind of cells? What kind of cells? Simple columnar epithelial cells. We also have a bunch of goblet cells in there too. What are those doing for me? You've seen this under the microscope. Remember way back when you looked at tissues and you looked at the small intestine and you looked at simple columnar epithelial cells? Remember that? And you saw the little bubbly looking things, kind of look almost like cracked glass in and amongst all of those other simple cells. Those goblet cells are going to produce what for me? Mucus. And why do I need mucus in the small intestine? Two reasons. protection and movement. I have to move things along. So we'll find a bunch of goblet cells dotted throughout there as well. Remember, in the submucosa, we're going to find things like mucosa-associated lymphoid tissue, Peyer's patches, or MALT for short. What's that? You just learned it in the lymphatic system. It's lymphatic tissue, so we're going to find cells there to help, because everything you eat is sterile, right? Totally sterilized and pure as the driven snow, right? No. So we have that there to help us fight infection. We also have some enteroendocrine cells down there at the bottom in what we call the intestinal crypts, not the gastric gland, because that's where? In the stomach. And those cells are going to help produce chemicals to set up who? Who's coming next after we do the small intestine thing? The large intestine. So, just like we saw previously when I talked about secretions from the stomach setting up the small intestine, those secretions from the small intestine are going to set up the large intestine. You guys following me, or you're all in a coma? You following me? Just no response today. Got the deer in the headlights going on big time out there in the crowd. So these secretions are going to help set up for the next part of the digestive process, which is the large intestine. So we call those intestinal crypts, or crypts of, I've never seen this term, Leberkuhn. Leberkuhn? Leprechaun. I don't know. Anyway, kind of like a German Scottish guy, I don't know. He's the one that figured that one out. The other thing I want to point out about this lining is not only do we increase surface area by creating these things I can see with my eyes called villi, but the cells themselves, and that's what that little picture up at the top corner is, those are the cells, the simple columnar epithelial cells, that also have little finger-like projections on them to even further increase surface area in the small intestine. Those microvilli, very small little villi, they're not cilia. There's a difference. Cilia was what? Yeah, almost like a hair-like projection. We had a bunch of microtubules in there, and actually there's like a little protein motor at the end of them that cause them to wave back and forth. That's not these guys. This is just continued increased surface area of the plasma membrane. So they're microvilli, and because if I look at them under the microscope, if I have a really good microscope, they kind of look like a brush. So what do we call the lining? The brush border. These are the cells that are also going to produce what? The brush border enzymes. So that's where they got their name, because of their looks. And this should look a little bit familiar, much different stain, but we see the goblet cells in kind of a yellowish at the top. The villi themselves are the finger-like projections, and we can see this when we look inside the small intestine. So another thing, if you get to open up your small intestines, have a look. You can actually see them. And then we have the absorptive cells with the brush border of microvilli near the top, where absorption is going to take place. In this particular diagram, that green portion is where I'm going to find my blood vessels, and that's the submucosa. So here's another peek at some villi, and a look at the microvilli. Again, this is a cell, an absorptive cell, and the little microvilli on top of that. So that's the brush border, right? So that's the histology of the cell wall of the small intestine. Now we're going to talk about, we talked about Peyer's patches. What's a duodenal gland? Making some mucus. Mucus is going to help me move things along. What's the composition of intestinal juices? Well, we're going to have a whole bunch of different things. And that diagram that you're supposed to know for your lab exam today, give me some examples of what we find in intestinal juices. Some of those brush border enzymes. Trypsin, maltase, sucrase, lactase. Is that ringing a bell? What are they going to help me do? Break down who? Go look on page 896. Figure 23.32. Yeah, break down carbohydrates. We need to break down amino acids with peptidase, pepsidogen, all that wonderful stuff. We need to break down amino acids with aminopeptidases. Amino acids, remember, were the building blocks of proteins. Carboxypeptidases. We need to break down carbohydrates. How about lipids? Who's going to help me break down lipids? Yep, so lipase from the pancreas and also small intestine. Ribonucleases are going to break down deoxyribonucleic acid or nucleic acids. You eat nucleic acids? Hell yeah. Name me something you eat that's not alive or hasn't been at one point. Yep, you do eat dirt. Not on purpose, but you do eat dirt. I'm thinking things you eat on purpose. Yeah, salt. Good. That's about it. Right? Because everything, even your spices, if you throw some basil or oregano or whatever on your food, was at one time a what? It was a plant, a living thing with what? DNA and RNA. And what are you going to do with that? Break it down into its building blocks, its nucleotides. Remember that way back when in the chemistry chapter? And what are you going to use them for? To make your own DNA and RNA so that you can go through transcription and translation. See how all this crap works together? You're like, I'm never going to have to remember that. Yes, you are. Because it all comes together in second semester. So first semester that haunted you, here it is again to haunt you again. All right? So all of those things are building blocks for what you need for your chemical process of metabolism. And that those enzymes have to be produced by different portions of the digestive system to help you break those things down so you can use them. So you can do what we learned in chapter, what the heck was it, chapter 2 or chapter 3? And what we're going to learn when we talk about cellular respiration, and you're going to love that. And what's cellular respiration for? To create who? ATP, adenosine triphosphate. That's our energy molecule. So that big, huge chemical reaction of metabolism, which you're going to learn as well in this class, involves the building blocks that we get from the digestive process. See how everybody kind of works hand in hand? So you remember that expression, you are what you eat? Yep. Because you get your building blocks for all of these reactions of metabolism from what? Powdered little donuts. So you are a powdered donut. Oh, and coffee. Okay? So, what you eat is what you are. Think about that. Every once in a while, you know those little green things, the fruits, the vegetables? You might want to throw those into the diet every once in a while. because those give you very important building blocks in the digestive process for metabolism. So remember the story of the 3 pigs? Remember the 3 pigs? The straw house. I wanted to make sure I only had 3 fingers up. I'm tired. Straw house, stick house, brick house. You want to be a brick house. So you want good strong mortar for your building. You give yourself shoddy building materials, you make a shoddy building. Because that's all you have to work with. Yes? So, just throwing that out there. That's an inside, just in case you need another science class. You can take my nutrition class spring semester next year. So, those are intestinal juices, and again, that diagram, and you having to memorize it for your lab exam, hopefully you've already done that this week, gives you a good idea of what the composition is. The secretions from the pancreas are also going to have one added little extra thing beside the enzymes that we talk about. What do you think it is? One of the things we kind of lost track of is, remember how acidic the secretions were when things dumped in from the stomach into the small intestine? And I said we have to be careful because the small intestine doesn't like it that acidic. Bile's going to come from the liver. But one of the other things that the pancreas is going to help me do is secrete more hydroxide ions, more of a basic type secretion to do what to the pH of chyme. Bring it back up to that more 7-ish range, because that's where we want to be in the small intestine. So secretions from the pancreas are also going to add sort of an alkaline substance to the chyme to bring that pH closer to that 7-ish range where we want to be. So that's another thing we're going to find in the composition of intestinal juices in the small intestine. All right, gross anatomy of the liver. Big, huge thing. Where's your liver? There's your liver. Whole bunch of lobes, kind of plastered to the underside of your diaphragm. It's quite a big organ. It does a lot. It has many jobs. It's got to make bile, it's got to filter blood, it's got to get rid of old red blood cells. We've talked about it several times already, haven't we? So there's quite a bit of tissue there, and the liver is a very large organ. What's this? Ligamentum teres. You should know that for your lab exam, too. Circulatory system's in your lab exam, isn't it? What is that? I'm giving you a hint. Huh? It's what? It's a navel? No. I'm giving you a hint, though. It's the remnants of what? The umbilical vein, umbilical artery. Your connection to mom. You're a mammal, right? Your mother did all the work for you while you were developing, including, we talked about the circulatory process, but what else? Yeah, the whole digestion thing. She had to do that for you before you were born, too. So where did the nutrients then feed in? To your hookup to mom, the umbilical artery, umbilical vein, that whole setup in the umbilicus, yes? And then when you get born, it's probably not the proper anatomy term, but when you get born and they cut the umbilical cord, what happens to that connection to mom? Kind of shrivels up and turns into a ligament that helps to hold that big, huge liver organ in place. So it kind of, it still holds it down a little bit, kind of connected to your belly button. Does that make sense? Kind of? Seeing. I'm pretty sure that was a homework question. Yeah. Yeah. What that was. Yes? No? Hopefully you did your homework. You had to go find it. So anyway, that's where it is. So the liver, big, huge organ that's going to be the accessory organ to the digestive process. And that wonderful little gallbladder, the place where I'm going to store and concentrate bile, is kind of plastered to the underside of, excuse me, the liver. We also, in humans, we're not going to see them in the pigs as much, is we have a bunch of membranes that kind of act like they almost look like an apron when you dissect a human. They're called the omentums. And they're serous membranes that kind of flop over everything in the abdominal pelvic cavity. Again, those serous membranes are going to produce fluid to help keep everybody from rubbing against each other and creating friction. But the other thing that we're going to find in the omentums, and we can see a little bit talked about there up at the top, and that's why I mentioned it, the lesser omentum, we're also going to see little fat tabs. Now we know why, especially women have that little issue with fat tabs. Yes, because they're in our omentums and they fill up. And what are those fat tabs there for? Especially in women. Who are we trying to protect? Yeah, developing fetus or embryo if we possibly have one of those. So women tend to accumulate more fat because we have more fat tabs for protection in that region. They were also to provide nutrients for who? The organs in the abdominal pelvic cavity as well. So they're important. Unfortunately, sometimes they can get a little carried away and fill up too much. So we looked at this under the microscope. This is the setup of the liver. And if you look on page 883, we see the nice illustration of this. So we have a bunch of different cells there. Again, some of those cells are going to make bile for us, and those are the guys we're going to talk about a little bit more in this chapter. They make bile for us and then shoot it down little areas in these zones. And what are these little zones called? They're called lobules. And lobules are made up of a bunch of cells and kind of outlined by blood vessels and lymphatic vessels and bile-carrying vessels called bile ducts. These vessels altogether are called the portal triad. And again, they consist of bile-carrying tubes or bile ducts, portal venules, and portal arterioles, so arteries and veins. Okay? Oops. It's all three of them together. And you can see them down here. But that's what's going to kind of surround these lobules, all of these tubes. They're going to feed into larger portal systems. And the hepatic portal system in general is a very, I can't think of the word I want to say. A whole bunch of tubes. What do I want to say? Can't think of it. A whole bunch of tubes that feed into each other. Very elaborate, that's the word I want. A very elaborate system of tubes in the hepatic portal system. We didn't go over that specifically when we did the circulatory system, but if you go back and look in that chapter, you can see, remember how we kind of skipped that part in the veins and arteries chapter at the end? If you go take a peek at the hepatic portal system, you can see how complex and elaborate it is. But this is the basis of it. So we see a bunch of little bile ducts, and that's what we're going to talk about when we talk about digestion. Those are going to feed into the gallbladder. Now, some of the cells that we're going to look at are cells, and we can see these are the pancreas, so they don't really go into the... Well, they talk a little bit about Kupffer cells. You don't really get crazy about, oh, there we go, hepatic macrophages on page 881 under the microscopic anatomy of the liver. These are some of the cells that make up the lobules of the liver. So Kupffer cells are macrophages, and they're going to help clean out debris. If there's any bacteria, worn out cells, red cells especially, we talked about them when we talked about the reproduction of the recycling of some of the materials in red cells, and the liver was in play there. So the portal triad's going to feed all of that stuff through those little lobule setups in the liver. And the lobules are basically made of the different cells we're going to find in the liver. Can't talk today. All right, so homeostatic imbalances, anything that damages movement of materials through the liver can be a big problem. Hepatitis is a virus that attacks the cells in the liver and can cause problems with liver function. Again, liver function is very important. It's kind of the garbage dump of your body. Gets rid of old things, recycles things, and of course is important for the digestive process. So hepatitis is tough. Cirrhosis they talk about here as well. Anybody know what cirrhosis is? It's one of the complications of alcoholism, but it's basically like scar tissue forming in your liver. So cells becoming damaged and no longer being able to function, doing all of that garbage taken care of that they normally do. Remember, they convert bilirubin into bilirubin and all of that stuff we talked about when we talked about red blood cells. Well, that byproduct is kind of yellowish. So one of the things you can see in patients who have impaired liver function is they'll start to build up that yellowish waste that the liver normally took care of in their blood plasma. So if you draw a tube of their blood is their plasma. Remember how we looked at it in lab was kind of a straw colored? It's yellow because of all those byproducts. In turn, your skin starts to look yellow. Excuse me. Because of that in your blood plasma. Yeah, and that's another job of the liver is to help balance out fluid in the abdominal pelvic cavity, so you can have fluid accumulation. That's called ascites. And even the whites of your eyes will no longer be white. They'll be yellow. So those are some of the things that you can see in patients with impaired liver function, whether it be cirrhosis or sometimes hepatitis patients. Again, because of impaired liver function, will show those symptoms as well. They talk about composition of bile. Bile salts are going to be emulsifiers. They're kind of like, almost like soap, to kind of break apart fats, not break them down. Who's going to do that again? Lipase. Bilirubin is also a component of bile, and that was from the breakdown of old red blood cells, so that's going to help as well. They talk a little bit about the gallbladder. Remember, gallbladder concentrates bile, so sometimes little stones can form or crystals can form, and they talk about that under homeostatic imbalances and gallstones. All right, now we're into the pancreas, another very important exocrine/endocrine gland in your system. We've already talked about the pancreas, haven't we? When? No, before. Now we're talking about breakdown in the small intestine. But when did we talk about the pancreas before? In the endocrine system. And what was its function? What did we talk about? The islets of Langerhans. Again with the Germans. Nobody? Alpha, beta cells. Insulin and glucagon. Beta cells produce insulin. Insulin's going to help you do what? Regulate blood sugar. Take that glucose from your plasma and do what? Suck it into your cells. When I don't have enough glucose in the body, who's going to help me make sure I get it? Glucagon, released by the pancreas as well. So we already talked about this guy. Now here's its digestive function. So we have a bunch of cells that we're going to find in an area of the pancreas called Akinner cells. And we saw them already when we looked at the pancreas under the microscope. Do you remember labeling them? Does that ring a bell, this Akinner cell thing? And what are those doing for me? Yeah, enzymes. Enzymes that are going to help me with the digestive process. So if you look on page 886, you will see the composition of pancreatic juice. So we're going to find things like trypsin and enteropeptidases, chymotrypsin, carboxypeptidases, amylase, lipase, nucleases. What's amylase for? Carbohydrate break apart. What's lipase for? Lipid breakdown. What are nucleases for? Nucleic acids, DNA, RNA, break them apart into nucleotides. So those are some of the secretions we're going to see from the, the pancreas. And those again are going to go out the common bile duct into the small intestine. So we see some of the chemicals listed here. Trypsinogen, chymotrypsinogen, carboxypeptidases, and again, when they come in contact with some of the acids from the chyme, they are going to activate these guys and start them being able to break down some of the substances mentioned. Oh, I'm tired of talking. You tired of listening to me? All right, so we'll pick up with this on, and we'll definitely finish chapter 23 and pretty much get into chapter 24 on Thursday. No, I forgot the what's this today. You get a free attendance today.
I'm going to be missing